Hello and welcome to this webinar titled Beyond Purely Data-Driven Approaches for Efficient Knowledge Management in Process Development, hosted by Biopharma Asia magazine and presented by Moritz von Stosch, PhD, Senior Manager, Technical R&D at GlaxoSmithKline Vaccines. This presentation will be followed by a presentation by Klaus Mauch, CEO at Insilico Biotechnology AG, and Shilpa Nargund, Data Scientist and Business Development Manager, also at Insilico Biotechnology AG. My name is Stephen Edwards, and I'll be your moderator. Before we begin, I would just like to inform our viewers that there will be a live Q&A session after both presentations near the end of the webinar. Audience members can send their questions in at any time during the webinar via the Questions tab located directly below your webinar screen, and we'll go through these at the end of the webinar. Now, please allow me to introduce our first presenter, Moritz von Stosch. Moritz works as Senior Manager at Technical R&D of GSK Biologicals, Belgium. He is a Process Systems Engineer by education with a diploma in Chemical Engineering from the RWTH Aachen University and a PhD in Biochemical Engineering from the University of Porto. Before joining GSK, Moritz worked as a lecturer at the School of Chemical Engineering and Advanced Materials at Newcastle University, where his research focused on the development of novel hybrid modeling methods and their application to enable more efficient process operation/design. I will now be handing over to our first presenter. Welcome, Dr. Stosch.
So, if we look at the current media coverage, what we'll see is that there's a large emphasis on man versus machine. And of course, if we look at certain games, then we'll see that in fact, when we have a man versus machine scenario, most of the times the machine wins. We saw this, for instance, back 2 years ago when there was the Go game, where the actual Go champion lost against the AlphaGo, which is a Google company, or we saw this for chess, where Garry Kasparov lost against Deep Blue back in the 90s, I believe. Now, learning from this, of course, rather than going against the machine, you would want to join up with the machine, and if you then play against another human, most likely you will win, right? But the much more interesting question is, what will happen if you join with a machine and you play against another machine? Now, it has been shown that for games such as chess or Go, if you join with a machine, in 60% of the times, the machine which plays together with you will win. And this is mostly due to the fact that you'll have some intuition about how to play the game, whereas the machine provides you with the computational power to really overpower the other machine. Now, my question for you is, why would we, when it comes to process development, try to go for an approach where it's man versus machine? And this is really what this webinar and with my talk will be all about. It's not man against machine, but in fact we should go beyond our data-driven focus for process development and integrate more and more knowledge. In fact, we should really look into how we can efficiently transfer the knowledge that we have from one project to another. As Stephen said, I'm working as a Senior Manager at Technical R&D Vaccines, GSK Vaccines here in Belgium. I'm leading the Process Systems Biology and Engineering Center of Excellence, and throughout my entire career, I've been focusing on developing methods that combine data-driven approaches with knowledge that we have from engineering, physical, chemical, or biological sciences. Before I go on, I just need to point out that, or disclose actually, that I'm an employee of the GSK Group, as you might have already imagined, and I'm also co-founder of Novasign, which is a company that provides commercial hybrid modeling software. Now, I want to show you why it is so important, in particular our area, that we go beyond the data focus. If we look across the industry and we look about the amount of data that is available, and the impact that we could generate from this data, and then if we look at, for instance, the well-known Googles and Facebooks and so forth, you can see that they have a lot of data available because every time you go browsing, you generate data. And of course, if they are capable to present you with more personalized advertisement, this means they are more likely to sell products. And so there's a high amount of data, but also the impact is fairly high they can generate. There's another area, which is the area of optimized pricing. And you might know this when you look at flight tickets. The more often you look at the same tickets, the more likely it is that the price increases. And that is because they know that you're willing to pay for this flight, and therefore they increase the price. Optimized pricing is all about that. They know about your personal background, they know about your wish to buy a certain product, and therefore they adapt the prices based on your wishes. High impact, relatively high amount of data, because every time we buy something, we in fact generate data. There's another area, which is the area of personalized workout. We currently see all these devices, such as watches or so, popping up where you can record your workout. Of course, based on the recorded workout, the machine can help you to direct a personalized workout program. And while there might be a lot of benefit to you, at this moment, there's not a lot of economic value which is generated from these data. Now, where are we in the bioprocess industry? We are somewhere down here, right? There's not a lot of data available, but if we could exploit that data that we have, we would have a large impact. Now, why are there not so many data available? This is because, in contrast to the other industries, in our industry, it's extremely expensive to generate data. And so we really need to ask ourselves, how can we leverage the data that we have to the maximum amount possible? And that's what all this talk should be about. Because in fact, what we have in our industry, what we do not have in the other industries, is other domain knowledge. We might know a little bit of what happens if you work out, right? We have been studying this for ages. We have a notion of what personalized marketing could do, but we don't really understand from a fundamental point of view how it works. And the same is true from optimized pricing, even so there's a lot of theories out there. In our industry, we have a lot of knowledge available. There's a lot of knowledge from physical, chemical, and biological sciences, and we could readily integrate this into our process modeling approaches and, in fact, combine this with the data that we have to leverage the pure and small amount of data that we have to a much larger extent. Let me now come to the outline of my talk. What I want to start with is the Quality by Design initiative, and that is coming from the roadmap that you see here. In fact, everything that is blue here is product related. So we start with a target product profile. We go into the identification of the critical quality attributes using some risk assessment. Then we define the product design space, again using risk assessment. And then once we have the product attributes defined, we go using risk assessment into our processes and we see, okay, what part of the process do we need to characterize and what part of the process potentially doesn't have a huge impact. And we then go from the process characterization, which is mostly done using design of experiment and statistical tools, towards defining a process design space, go forward with defining a control strategy, validate the process that we developed, and then we suggest process monitoring strategies in order to make sure that we see or see not that the process is on track. Now, as I said, when we do this, mostly we're really using design of experiment and statistical approaches to go all the way from the process characterization to the monitoring approach. But what we have and what we could easily integrate is all the biochemical engineering knowledge that we've been taught during our education. So the first part of my talk and the first case study will focus on how we can leverage our knowledge in one particular project, which will be a E.coli upstream process. Now, once we've introduced our knowledge for one particular project, we can of course ask ourselves, is there not a better way we can leverage the knowledge and really transfer it from project A to project C to project D and so forth? And this is what the second part of the talk will be about. That is, how can we transfer process knowledge across projects and how can we ultimately manage our knowledge in a much better fashion? Let me now come to the first part of the talk, how to use process knowledge, engineering process knowledge, much more effective. Now, as I said, we have a E. coli case study here. This is a typical drug substance process. It's the production of a recombinant protein in a fed batch process. And what we have done, and this is really before I came in, we have done a DULA design, which is a particular design of experiment. And then we have, based on these data, generated a response surface model because we wanted to understand what is the impact of temperature, pH, the initial biomass concentration, and the carbon substrate feeding rate on the final product. Now, what we've done is that we have executed all these experiments at the different levels, and then we recorded the product at the end time. We could then derive the response surface model, and from the response surface model we found the optimal conditions that maximize product titer, and we could also see in repetitions that we have very little variability at the optimal process conditions, and this means that we really have a robust process. Now, if you look at the experiments that we have done, you can see that in line 1 we started with a design where we did systematic changes in all 4 factors, and in fact we started with 23 experiments. Now, what we saw after we have analyzed the experimental data is that in fact the optimum was not where we investigated. So we had to shift our experiments slightly, and in order to shift it in the direction where we presumed the optimum was, we did some investigative batches, and in fact then we run a second experiment study, and that is line 5, where we did 8 additional experiments. Now, what we saw is that rather than where we expected the optimum to be, and we're still not there, and so we shifted the design again. We did another 8 experiments. We did investigative batches to be sure that the optimum is the optimum, and then we did 4 experiments to study reproducibility. So throughout this project, we had done 53 experiments, and this is 20-liter experiments, right? It's not like we have done those in an amber, otherwise it would be fairly cheap. This is when I came in and I said, okay, now we have all this data and we have done our statistical approaches, but why have we done this in that way, and why do we not leverage what we know about the process in a much better fashion? And without any knowledge about the process data, I've just written down the material balances, and this might look awfully complicated, but I'll walk you through it and it's fairly easy. So in the first line, what you see is the material balance of biomass. This dx over dt means this is how biomass changes over time. It's small incremental changes in biomass, dx, over small incremental changes in time, dt. And what you see on the right-hand side, the first term is the growth term. We have mu, which is the specific biomass growth. X is again biomass, and then on the right-hand side we have the dilution term, which means when we add material to the reactor, and of course we have a certain amount of biomass in there, then it will be diluted just by the fact that we add volume, and this is what this D stands for. Now the second line is essentially the same for product. We have here some more complicated terms, so this whole term in the bracket is derived from a consideration that in fact we should look at specific product, that is this product which builds inside the E. coli, because the E. coli is not secreting the product, but the product is in fact accumulating inside our organism. And therefore we have these slightly more complicated terms in the bracket, but again it can be fairly easy derived using some considerations. Then on the right hand side you will see the integral of U base times dt, and then this equals some yield times the change in total mass of biomass. And this equation in fact is the material balance for carbon dioxide in a simplified form. And what this says is that when we see a change in biomass, then this change comes typically with a change in the base that we need to add in order to control pH. And why is that? Of course, if biomass grows, it breathes and it produces carbon dioxide. This is dissolved in the medium, and because then we have acidification, we need to add base in order to keep the pH constant. Now this yield, YBX, this will be changing with changes in temperature, with changes in pH, and so forth. And because we don't know how this really works, we have this delta YBC down here, and we need to model this in some way. The same is true for the specific biomass growth, this mu, and also the specific product formation rate, which is this VBX. So these three functions, we don't know how they look mechanistically or from first principles, but in fact we have to derive those from data. And this is exactly what we did. We put in a neural network to model us how nu, VBX, and delta YBC evolve as functions of our biomass, our specific product, and our factors, namely temperature, pH, and the carbon substrate feeding rate. And by doing this, we combine what we know with the data by just plugging in what we don't know, right? And we represent this knowledge that we don't have by a neural network. It could also be another machine learning approach. We just show the neural network here because it's nonlinear and it's a fairly complex problem. Now, when we derived this model, we then went on and we used the first 23 experiments to just develop the model on this. And what you can see here are the predictions over the experimental measures for biomass, then specific product, then in the lower plots product, and the base biomass growth correlation. In red are the experimental data and the training data that we used to develop the model. In blue are the data that we used to validate the model, and in green are the data that we used to test the model. What you can see is that, in fact, the predictions or the estimations and the experimental values align very, very well. And that is true for biomass, product, and the correlation. What we can also see is that there's a slight gap in the values, and that is, in fact, the gap when we have overnight measurements, because no one took the measurements, of course, overnight. Now, what we then also did was we developed the model, the same model, but using all of the data. So that is all of the 53 experiments that we had available. And again, in red, you see the training data, in blue, you see the validation data, and in green, the test data. Now, in this case, when we developed the model on all the 53 experiments, and this is what I'll later refer to as Model 2, we can see that for product, we have slight mismatches in high product regions. So we see, in fact, that the model is not capable to predict in certain regions of our product space. Now, when we looked at that, we looked at the process conditions that we had at this particular settings, and we found that the organism is likely to go into an overflow metabolism in these conditions. Now, an overflow metabolism is typically characterized by the production of byproduct, but because we did not measure nor model byproduct, the model doesn't really see that it goes into an overflow metabolism, and this is why it cannot represent this state accurately. However, we're not really interested in this region because in this region the process is not stable. In fact, we see switches in the process. And because we're not interested in this region, we were not utterly concerned with the mispredictions that we had in this region. Now, what we looked into next is we compared the model performances of the two models on different settings of the experiment. And what you see here is 4 experiments of our design space region 1. In this region, you see on the left-hand side biomass, on the right-hand side specific product. So biomass is in black, a specific product is in gray, and the dots are for the measured values, and the square dots are for biomass, the round dots are for a specific product. And what you can see is that both models, so one model is the dashed line, the other model is the continuous line, both models in fact predict accurately both biomass and specific product concentrations. Now, our question was very easy. Now that we predict accurately in the design space 1 or in the first investigated space, can we use the models to make much better predictions in the space 2? And what you see next is that when we use the models in the space where we find the optimum, we can make accurate predictions with the second model that we derived on all the 53 experiments, but we cannot make predictions with the model that we derived on the first experiments only. And that is because here the model has to extrapolate in temperature, in carbon substrate feeding rate, and pH. And because the weakest part of our model is the data-driven model, and because the data-driven model in this case has to extrapolate, of course it will not make good predictions, because it is known that data-driven models are weak when it comes to extrapolation capabilities. However, what is very interesting to notice is that if we look at our process, and this is really the concentrations over time for a specific product, we see that the maximum product concentration is not reached necessarily at the end. Rather, it is reached at about 30 hours. And what that means is that in principle, we could stop our process at 30 hours. We would gain 15 to 20 hours, depending on how long we want to run our process. And in this 15 to 20 hours, we could do something else. And that essentially means that without any additional experiments, we have gained knowledge of how long we should run the process. And in fact, we can also show that we can gain knowledge of when we should induce the process. So this is two additional factors, namely the total amount of process, the total process time, and the time at which we should induce, which we have gained without doing any experiments, just from the experiments that we have here. And this is because we have, of course, introduced the knowledge into our model. What we were concerned with next is what can we do with the model. And we looked at, can we make decisions on what happens if we, for instance, have a pump failure? And that means we really look at the dynamics, because one particularity of the model that we are investigating here is that we can make much better decisions of what happens to the process if we have variations in temperature, feeding rate, or pH. And what you can see here is for different moments and times, how specific biomass growth and how specific productivity are impacted by changes in pH, the carbon feeding rate, and temperature. And you can see in particular for product that this profile is highly nonlinear, so it's rather difficult really to make a good statement of what will happen just from the instinct that you might have. Now, when I showed these particular plots to the people at GSK, they were very keen to see whether this is really true, and what we designed next was experiments to see whether we can really make predictions from these experiments, because all the experiments that we had done to this date were static, right? They were at completely static temperature, pH, and carbon substrate feeding rate settings. And now, based on the static settings, we make dynamic predictions. And we were not totally confident that this is really possible. So what we did next is we did experiments where we systematically varied. We did step changes in the factors, so in temperature, in pH, and in the carbon substrate feeding rate. And what you can see on the bottom are these changes in the factors, and on the top you can see on the left hand biomass, and on the right hand you see the specific product. In black you see the concentrations and the measured concentrations and the prediction by the two models. And in gray, in fact, you see the specific rates, the prediction of specific rates. And what you can see very well is that if we change the process conditions, then the model indicates, in fact, that there is a change in the specific growth rate. And it can, even though we changed something, still predict accurately how the concentrations will evolve. And this is really interesting, because that also means that in principle we could pack our original design of experiment into a dynamic, or as we term it, intensified design of experiment, run much less experiments but with a dynamic setting, and gain the same information. And this is what we investigated next, but which I will not show here for the sake of time. If you're interested, you can get to the publications, which I'll show in the end. Now, what did we learn during this study? What we learned is that we can use the hybrid model to accurately capture the impact of the control degrees of freedom of our critical process parameters on the process, namely those of temperature, pH, the carbon substrate feeding rate, and the initial biomass concentration. Furthermore, we had gained 2 additional control degrees of freedom without doing any additional experiments, and that is process time and the induction time. And then, of course, this model allowed us to make really accurate statements on what are the dependencies of the specific rates on changes in the control degrees of freedom during time. So like a pump failure or a failure in the heating, what is the impact of this on our process? Now, summing the first part up and going to the next part, this was really project specific. And what I want to assess now is how can we transfer this knowledge that we have gained from one project to the next. And I want to show you this on a controlled drug release case. So what we have here is a nanoparticle or a microparticle. And this microparticle is loaded with a drug. And in fact, it's a drug polymer combination. And then this drug polymer combination is injected into humans. And then the drug is supposed to be slowly released from the human in a really sustained release. And what we want to avoid is a seesaw effect. So that means that we have quick release, then slow release, quick release, slow release. And we want to avoid this because we might reach toxic concentrations, or we might have concentrations that no longer impact on the particular disease we're targeting. So I had a PhD student doing that, and what she did was she collected over 150 experiments for 40-something different drugs. And we saw that for these 150 experiments, the initial release profile, the burst release as we call it, always looked something like this, which is an exponential function. And in fact, there was a publication out there which had found this very simple equation where the accumulated release Q is essentially modeled as a function of this F-bin factor, which is the total amount of material released during burst, and then this time-varying function with an exponential and Kb. And we can now, by using these two parameters, F-bin and Kb, really modulate the response. So the higher we go in F-bin, the more amount is released, and the higher we go in Kb, the faster the release is. So we saw that this relationship holds across the different factors, but in fact F-bin, so this one parameter, and Kb, they are different from experiment to experiment, and they really depend on the different properties that we have when we synthesize our particles, and also they depend on the drug and the formulation characteristics. So for these 150 experiments, what the PhD student did was she collected all the data on the drug molecular descriptors, on the experimental conditions, and on the formulation characteristics. And what she did next was that she linked the parameters in this equation that we had to these different settings in the experiments. That means that for every setting, we could predict what the Kb and F-bin was, and then using this equation, we could calculate what the cumulative release should look like, the cumulative release over time. Now, we did a bootstrap aggregating method here, and in blue, you can see the train and validation set. It's again the predicted quantity over the measured quantity, and you can see that the training and validation works fairly well. And then we had a test case, and you see that there is some fair errors in the test case, in particular in the lower part, but this is due to the fact that we have in this test case very large proteins, and these very large proteins were not fairly represented in the training and validation set, so it's no wonder that we are mispredicting in this case. However, overall, the prediction capabilities is somewhat acceptable, and it's much better to predict what settings we should look like than to go by trial and error and figure out what these settings are. So this really means that we now developed a model where we can relate the drug, the synthesis parameters, and the particle composition, the formulation characteristics. We can relate those properties to the amount of burst, and we can also relate those to the burst kinetics. And now we can go the way back, meaning we can, for a completely new drug, unseen before, and for desired burst kinetics and for a desired amount of burst, for these properties we can design what the particle should look like, and we can define what the synthesis parameters are. And we have done that, in fact. So what I show you next is a study where for a new protein, we have looked at how changes in our parameters impact on the accumulated amount of drug that is released over time. And you can see that for most of the parameters, there's a significant impact on the burst release, except for this PVA inner concentration where the release profile stays fairly flat no matter what the parameter is. But that means that now really in silico, you can understand what happens if I change my experimental parameters, what happens if I change the particle design, what happens with my accumulated amount of drug released. So what we've seen in this project is that we can transfer this knowledge equation that we have from one project to the next, and we can in fact use the data to understand what is the impact of changing conditions on our overall response. And that means that really we reuse what we know, and in a much more effective way we can now design the drug carrier system, and we can now rationally design the experiments. Now, what was fairly interesting to us was that we could in principle add to this data that we already had also patient data, and we could then, for instance, for heavier patients or less heavy patients, really tailor the design of the nanoparticle to the properties of the patient. And that means that we ultimately go one step further towards personalized medicine, and in this particular case it would be, of course, personalized design of the carrier, so of this particle which loads the drug. And that was totally mind-blowing for me. Now I want to wrap it overall up by saying, when we look at the knowledge pyramid and we look at the bottom where we have data, and we look at the top where we have first principles, it should be clear that we should refrain from only using data in our projects. We should really take a holistic approach and integrate all kinds of knowledge that we have into a coherent model that serves the purpose. Because more process data does not mean more process knowledge. In fact, if most of the process data is redundant, there's no additional knowledge that you might gain. However, if you use your first principles right, and if you use your rest of the knowledge right, you can reduce the amount of data that you have to generate. So what I'm really advertising for and advocating for is to integrate more of the knowledge into our projects, into our discovery, development, and lifecycle projects. And by doing this, I think there's a tremendous opportunity to reduce timelines and also to reduce workloads. The other point I really want to advocate for is that if we do this, and if we not only use data but we also use knowledge, then overall we have a much more efficient process of knowledge management. And of course this means that we also, when we build our digital and data strategies, we also need to consider of how we really want to integrate all the other knowledge sources into our overall processes. Because if we neglect them, there might be a lot of value that we just leave on the way. Let me finish by acknowledging a lot of people. So first of all, I want to acknowledge Jan-Martin Hammelink, which was at that time the person at GSK who did most of the experiments and who really approached me and looked into all the different opportunities, and it was an amazing work with him, and we continued the initial study which I've shown here by developing an intensified design of experiment approach, which we published in this first publication. I also want to acknowledge Rui Oliveira, who was my supervisor during the PhD and also during my postdoc, and with him I developed all the algorithms which were used to do the work and which are now commercialized by Novasign. I want to acknowledge Mark Willis, with whom I worked a lot on the intensified design of experiment approach as well, but who had a significant impact also on the second case study, namely the controlled drug release study. And then the PhD student, which is Cristiana Maria de Azevedo. She has been sitting in the first 2 years of her PhD just gathering and cleaning and putting data together to really then be capable to make this dedicated controlled drug release study. With this, I would like to thank you, and I'm afterwards really happy to take your questions.
Thank you, Dr. Stosch. Now, before I introduce our next presenter, Dr. Mauch and Dr. Nargund, once again, I would like to remind our viewers that there will be a live question and answer session at the end of the webinar. Please feel free to send in your questions at any time during the webinar via the Questions tab located directly below your webinar screen. And we'll go through these at the end of the presentation. Now, please allow me to introduce our second speakers, Klaus Mauch and Shilpa Nargund. Klaus Mauch is one of Insilico's co-founders and became Managing Director in 2004 and CEO in 2006. In his previous role as CTO, he was responsible for the design and development of Insilico's modeling and simulation platform, where he established novel methods for the computer-aided construction of cellular networks. Before that, he gained extensive experience in metabolic engineering and applied systems biology as a group leader at the Institute of Biochemical Engineering, University of Stuttgart. He was awarded a master's degree in chemical engineering by the Technical University of Karlsruhe, Germany. Klaus is a member of the advisory board of the Division System Biology and Synthetic Biology of DESHIMA, board member of the Industrial Association White Biotechnology, and board member of the Association of German Biotechnology Companies. And Shilpa Nargund is a data scientist and a business development manager at Insilico Biotechnology AG. As a data scientist, she works on optimizing cell culture processes using Insilico's digital twins, whereas in the role of business development manager, she is developing Insilico's business in the APAC region, starting with the establishment of a Singapore office. Previously, she spent 5 years in early and late-stage cell culture process development teams in biopharma companies across the USA, India, and Singapore. She is a chemical engineer with a bachelor's from the Institute of Chemical Technology, Mumbai, India, and a PhD from the University of Maryland College Park, USA. I will now be handing over to our second presenters. Welcome, Klaus Mauch and Shilpa Nargund.
Thank you, Stephen, for this very kind introduction, and thank you guys for joining this session today. So we are going to outline actually our business very briefly, and we'll then shift gears towards presenting to you what's inside our Insilico Digital Twins, and you will see that we are following a hybrid approach that really goes beyond actually purely data-driven strategies. And we are going to show you application examples covering all, or the largest part actually of the process development task that we are encountering today. And I would like to close up by outlining how we are going to deal these problems from a computational point of view, and having a summary at the very end of this talk today. So Insilico Biotechnology is a company that develops and delivers these predictive digital twins that serves as a decision support for advancing biopharmaceutical process development and manufacturing. So we are doing at Insilico, we're doing consulting as well as technology transfer. So these digital twins that we are using in-house are also available on-premise and via cloud solutions. So if you look inside our hybrid Insilico Digital Twins, then we see on the left-hand side that we make massive use of knowledge that is already out there, and Moritz fantastically explained how we can use this knowledge to leverage our data and really go beyond actually the space where we gain the data so such that we can extrapolate and move this knowledge and these findings across projects and across scales. So what we do actually is we make use of the conservation of mass. We are using also known mass transfer kinetics. For example, we are making use of the oxygen transfer. We are making use of the sequence genome that is available for mammalians as well as for microbial systems. And we actually make use of the biochemical pathways that we now have at our hands. And this is going to be linked to the extracellular space where also important processes happen, like for example the oxidation of products and other quality relevant processes. So the digital twin is set up actually by this cell model, which is then actually driven by artificial neural networks in a way that we can integrate the known unknowns, which we still have a lot of, and which we are going to bring in by a data-driven approach. So what we still do not know very well are the enzyme kinetics, the parameters that are involved in enzyme kinetics, and that are in the end form the pathways that we have in our cells. We don't know the mechanistic effect of temperature and pH. We still don't know the mechanistic effects of trace metals. Osmolality is also something that where we can only use partially mechanistic approaches. And also importantly, we actually do not know how or the dynamics or mechanistics of the gene expression that we encounter when we look at processes such as the production of antibodies by mammalian cells. So these are features or effects are taken in by a data-driven approach and combined with a mechanistic approach within these digital twins. And if we again look at the data that is needed to train and to validate these digital twins, we heavily rely on actually time course data that we get most often from parallel cell cultivation. So this time course data involves cell viability, product titer, amino acid, lactate, ammonia, and trace metals. And typically we have actually one data point per day to give you an idea on the frequency of those measurements. But actually we need to know the concentration, feed media, the feeding volume, sampling time, all the stuff that can be measured easily or is documented anyway. And we're using that kind of data in combination with these known knowns to train and validate the twins, and to take these twins then to predict optimized platform media, optimized platform feeding, optimal clone selection, optimal scale up, to give you just a few examples. So these digital twins are applied along the value chain that we have, starting from cell line development to early stage process development and until manufacturing. So we are applying these digital twins for engineering cell lines, for doing clone selection, for doing media design, either for specific processes or for platform. The same holds for the feed design. We do apply these digital twins for process characterization, for process scale up, and finally also to lot-to-lot variation and to process control. And it's a pleasure actually having Shilpa here online, who will talk you through a couple of examples that show how predictive these digital twins can be. So Shilpa, will you take over?
Yes, thanks, Klaus, and hello everyone. I will go through the application of the Insilico Digital Twins. I will go through a total of 4 examples. So here's the first example of how we apply the digital twin, and this is mainly our bread and butter really, because this is what we do most often, that is feed and media design for optimizing product titer. So what I would like you to do is focus on the graph on the left. What we are showing here is on the y-axis we are looking at the titer, and on the x-axis we have looked at 3 different scenarios. So what happened in this case study is that the customer had a fed batch process for CHO cells, which had 2 feeds, and the objective for this project was to optimize the media and feeding scheme for increasing titer. But also it would have been nice if we can only optimize the feeding scheme and leave the media alone, because that is usually changing media composition is usually the more difficult task. So what we did was we looked at 3 different scenarios, which you see on the x-axis on this graph on the left. So we optimized the titer only for feeding scheme design, or for by changing or optimizing only the media, or by optimizing both. And you can see that optimizing both gives the highest titer, and it's also much higher than the reference titer, which is shown as the dashed blue line. And for this highest titer, we've shown what the changes in the feed media and the feeding volumes look like. So if you look at the top 2 graphs on the right, we're looking at the 2 feed medias, so feed media 1 and feed media 2. And what we see on the x-axis here are the media components, and on the y-axis the concentrations. So in blue you see the reference media compositions, and in red the optimized. The point I want to make on these graphs is that when we went back to the customer with this optimization, they said that it's very intuitive often to know what you should add to the media, because you see that's getting depleted in the spinatant, but it's very difficult to know what you need to remove in order to optimize it. And that is one way that the digital twin, which can do virtual experiments, can intelligently find out. On the bottom, if you look at the 2 graphs for the 2 different medias, what we're looking at is at the feeding volume strategy. So on the x-axis you have time, and on the y-axis is the volume. So the volume to be fed at this given time. In blue is the reference, and you can see that in the reference process, the customer had regular intervals or known intervals of time where given the same volume of feed was added. But what the digital twin was able to do was it could look at non-regular intervals of time, or whatever intervals of time that we tell it to look at, and it can figure out the exact volumes to feed. And you can imagine how difficult this would be to find out experimentally, because if you have to change both the feed volumes, the times, and also the feed compositions, it takes a large number of experiments. But this can be done far more easily using the digital twins, because you're running virtual experiments. Now let's see what the results look like when they were implemented. So the customer implemented this last scenario where both the media and the feeding scheme were optimized. Okay, so these are the results. So let's just go through the ledger first. The blue dots are the reference process. That's what the customer gave us to start off with. The red lines are the predictions and their uncertainty, the error ranges with the shaded area in red. And the green dots are the experimental implementation of the predicted process that we had given the customer. And you can see, if you focus on the graph at the left bottom corner, which shows the product titer, we can see the trajectories of the prediction and the experimental implementation very close to each other. And so this was a very successful implementation of what the digital twins had predicted. One take-home message I want to give on this slide is that you can see that the hybrid model can fit not only the product for which it was optimized, but also a lot of different curves for the biomass or your metabolites like lysine, valine, lactate, glucose, etc. And you see that all of these have very different dynamics. So they're all very different types of nonlinear functions. But the hybrid model is able to fit all of these pretty well. So this is a classic case of what we do on a routine basis, and we get to see what the dynamics, or we can predict the dynamics of what's happening outside the cells in the media to understand how we can control them. We can also look at what's happening inside the media. If I can move to the next. Okay. That went many. Okay, so we can also look at what happens inside the cells by doing flux analysis using the digital twin. What you're seeing here is just an example, just want to show you a part of flux maps that we create. It only shows you the TCA cycle and some associated pathways. And the point is that we can look at flux maps such as these at different time points. We're looking at a 48-hour time point, and we can understand what's going on inside the cells as well. So that wraps up the first example of how we apply the Insilico Digital Twins. And let's look at the next one. In this example, what the customer wanted was, again, they had a fed batch process for CHO cells that was producing IgG. What they wanted to increase the proportions of G1F and G2F in glycosylation profiles. So I'm showing you the implementation and the prediction and all of that together in these graphs. And if you look at the graph on the left, what you're seeing on the x-axis are the different major glycan species that were present on the IgG glycosylation. And on the y-axis, you have the percent of the total glycans as percent of a given glycan as a part of the totals in percentage. Here, the different bars represent, the blue one represents the reference process, so that's where the glycosylation started off at. The red is the predicted one, so this was predicted by optimizing the media for this process, and the green is what they got after implementing. So you can see that they achieved the G2F values, so the experimental implementation achieved the G2F values as was predicted. It fell a little short of the G1F values, but in general, the trends of shifting the glycan species from the usually undesired species such as G0F-N, MAN5, and G0 was accomplished. But this also came at the cost of a slight dip in the titer. So if you look at the graphs on the right side, we're showing biomass and product titer. So there was a trade-off between fitting or optimizing the glycosylation patterns against the titer, and this was something that was not captured by the model. But however, this trade-off was acceptable, and so that was okay for us. So this is the second example, and I'll move on to the next example. So here, this example is more of what I would call a case study, a use case, because this is something that we want to use to show or demonstrate how the Insilico Digital Twin can be used in process characterization. So we've just selected some critical quality attributes, and we've evaluated the CPPs to show what can happen. So let me just go through the details. So as you understand, the problem statement during process characterization is you have finished your process development, you've logged down your process, now you want to understand how robust it is. So you want to see if there is variation in any of the process parameters, how much does it affect one of your quality attributes. And this as you can imagine, this is one of the areas where virtual experimentation would be very helpful in finding out the good design spaces. So in this example, what we did was we chose titer as the critical quality attribute, and this titer was supposed to be within a 6.8 and 7.1 grams per liter. We did some preliminary analysis to find out that the interactions between asparagine and phenylalanine and asparagine and arginine are critical for the titer values. So we chose these as our CPPs. And then we varied these amino acid pairs within -20 and +20% of the original values to predict the response of titer. So if you will look at the graphs on the top, which are the surface plots of the design space, what you see is on the y-axis is the titer. On the x-axis are the percent variation in the individual amino acids. So if you have zero variation in both of them, you see the small black dot in the center, that's your original process or your lockdown process. And if you vary either of these amino acids plus or minus 20%, you see how the titer responds to that. So we have to go from here to a design or a control strategy. So we can use, for the same plots, we made contour plots. Now you can see the design space for these particular amino acids and the titer. So as I said, this is a use case, and this application can be discussed in a little more detail. But what I want to point out here on this in this slide is that previously the main limitation to finding out design space was the limitation on how many experiments you can do. But if you have a tool like the Insilico Digital Twin, which can do virtual experiments and basically navigate the entire design space, you have a lot more information. But now the challenge moves to the next step, which is when you have this much information, how do you go from there to the design, the control strategy? So that is something we are working on with our academic partners and also industrial partners. So that wraps up my example 3, and I will move to the next example. Here again, this is an example for clone selection. Again, this is a use case of how we can use our technology for clone selection. And as you all know, one of the main problems in clone selection is that how do we select clones that have a high potential for improvement during the process development phase? Because it's often not very easy to predict using preliminary data or early stage data on how much better your clone can get towards the end of process development. So that's the question we want to tackle using the Insilico Digital Twins. So what we would want to do is use the titer or whatever other product quality information we have from the early stage. And when we say early stage clone selection, it would be, say, well plate experiments, and predict what that clone will behave like at a future in a platform process or in a clone specific optimized process. So if you look at this graph, what we essentially want to show is that we can rank the clones. So on the x-axis you have the clone ID number, and on the y-axis you have titer. So if we ranked according to titer, but you can also rank according to several different criteria like specific productivity or the plasticity of the clone. You may find out that for a certain clone it would be much better to have a clone specific optimized process, and it would behave much better in that clone specific optimized process versus your platform process, and so you might take the call to do that clone specific optimization. And if you do that, of course, if you're doing that using the digital twin, then you are doing two things at the same time, that is clone selection and process development. So this is also a project that we are currently working with one of our customers, and hopefully we'll have good results very soon. So that wraps up the examples we have on how we apply the Insilico Digital Twins, and I'd like to hand back the presentation to Klaus.
Thank you, Shilpa, for this very nice overview. I would like to shift gears a bit by outlining how we actually can make use of these digital twins on a, let's say, scalable platform that everyone would like actually to have either in-house or as a cloud solution. So we thought that actually a solution that supports the digitalization and the usage of data should be highly scalable and should be designed in a modular way. So we are using microservices that are combined with standardized software like Google TensorFlow and IBM Cplex, Docker, just to name a few. and have these digital twins accessible by Jupyter notebooks. These are, let's say, lightweight web clients, and there the digital twins can be parameterized and tweaked in a way that those microservices are later on then combined in automated and standardized workflows, such that we have let's say, a fully automated twin running that provides decision support in an automated way. But in the first phase, we all work actually on this Jupyter notebooks and run and access this a server that has well, scalable hardware, such that this project can be conducted in a short time. So this is also, maybe if I can make this point, I think a game-changing moment that we see now, that these large companies, although they are working on much bigger data than we do, provide us with standardized platform solutions that we can readily use, which provides us and prevents us from designing and implementing algorithms. So nowadays we can make use of platforms like Google TensorFlow and other platforms, and that brings down the costs for creating digital twins tremendously. And from there we will also see some changing business models and some evolving new companies in the future. So if you have questions regarding this, and this goes clearly beyond the session today, please feel free to ask this by using the contact details we are giving to you at the very end of this session. So to sum up our Insilico hybrid digital twins, I'd like to actually mention that we were able to develop an ecosystem of predictive hybrid digital twins that can cover many parts of the upstream and well, also in the future also downstream manufacturing. These predictions that we get from these digital twins are pretty good. So actually we can really leverage the data that we obtain from the processes, and as outlined by Moritz, these are really costly data, and we can actually move these results to other projects, such that we can really leverage this kind of information by combining this data with mechanistic knowledge that we have nowadays. So we actually call these data that we use smart bioprocess data. We didn't talk too much about how we actually design this data, but we have actually to take care of how we also collect this data, the frequency, the accuracy of the data, the dynamics. They all have an impact on the predictive quality in the end. Well, and finally, I would like to mention that there's a good chance that by automating these workflows, by using data adapters, we end up with an automated fully automated solution that supports digitalization to a large extent. So if you have questions and comments that we cannot clarify in this Q&A session that we will run through now, please feel free to contact Shilpa directly at our Singapore office. And by this, I would like to hand back to Stephen.
Thank you for that presentation, Klaus Mauch and Shilpa Nargund. Now we will begin the question and answer segment of the webinar, but once again, I would like to remind the audience that you can still send your questions in via the questions tab located directly below your webinar screen. So now our first question is, given that pharma operates with process platforms, how much of the knowledge do you imagine can be reused in new products? And I believe that question is for you, Dr. Stosch.
Yes, so this is a very interesting question. I mean, in platform processes, of course, there's only minimal changes to the processes that we would like to do, and so I imagine that most of the knowledge that we have, in particular most of the mechanistic knowledge, can be reused from project to project in such process platform processes. So I would really imagine that most of the mechanistic knowledge can be reused, and then if we have a particular strategy in place, also our data can be leveraged to a much higher extent, as I hope I showed in the second part of the presentation.
Thank you. Our next question is, can historical data be used to train hybrid model? That's for you, Klaus and Dr. Nargund. Once again, that question was, can historical data be used to train the hybrid model?
I mean, thanks for that question. That depends on how the historical data is shaped. In principle, I would say yes. In many projects, this data can be used to come up with the first version of a digital twin that then is used for, let's say, an intensified quality by design and model-based quality by design, such that the predictive quality of this twin is enhanced. So if historical data is available, this actually facilitates the entry into this technology significantly.
Thank you. Our next question is, do you imagine that the integrated mechanistic knowledge could bias your analysis and modeling? And that's for you, Dr. Stosch.
Yes, so this is definitely a big concern. So whenever we integrate knowledge, we have to be very careful that the knowledge that we integrate is actually representing our process. So that is a particular problem if we're integrating, for instance, knowledge of metabolic networks, because it might be that we're constraining the solution space, and therefore we will not obtain a fit of our hybrid model to the experimental data, and we cannot obtain this fit. So whenever we integrate knowledge, we need to be very careful to which extent we believe this knowledge actually represents the process.
Thank you. And our next question. Why do you think these approaches only now finding their way into biopharma, whereas they are known already for such a long time? And that's again for you, Dr. Stosch.
Well, yes, so I started working on this back in 2007, and in fact from the very beginning I've always been promoting this approach and the use of this approach in biopharma. And I think it was particularly difficult in the beginning to overcome this skepticism of why these more complex approaches should deliver value. But I believe today we're in such a competitive landscape with the biosimilars and with many patent applications expiring that, in fact, we have to use these modern approaches to really leverage of what we know and to be more competitive and better competitive than if we just stand our way with traditional machine learning methods.
Moving on, the next question is it possible to fully automate the development and application of digital twins? And again, that question is for Klaus and Dr. Nargund.
So, well, I can take that one. So already outlined the vision for that. So the crucial point is actually the connection of the usually heterogeneous data world that we now have at biopharmaceutical companies to these workflows. So the data extraction, transformation, loading, and then actually the check curation of the data is actually the crucial point. If the data is generated by machines, we can actually do that in an automated way, and it is increasingly provided by machines. You know all that. Still, we have, let's say, offline measurements, at-line measurements, where still a lot of, let's say, manual steps are involved. But these will be replaced, and we see that process in many companies. So I'm very optimistic that due to these various digitalization strategies, we can actually derive and convey and process the data in an automated and standardized and defined way. That opens up the way to fully automated workflows.
Thank you. And on the back of that question, how does the development of digital twins fit into the overall digital transformation of biopharma? And again, that's for you, Klaus and Dr.
Yeah, so I mean, it's and Moritz also commented on that. It's actually crucial that that companies have to be more flexible. They have to be more productive. So bringing down actually the time for process development is crucial. So flexibility and time is really relevant. Plus, then the cost of goods is something we have to have an eye on, not only because of the biosimilars, it's also because in developing countries such as China and India and other countries, companies are trying their best to bring down development costs of antibodies from a range of today in the range of $100 per gram to a range of, let's say, $10 per gram, and then we are in a completely different game, because these processes will then compete also with processes developed in countries like the US and Europe. So actually, I would say that these predictive technologies can be and will be key to further process intensification and also bringing down actually the timeline. So it's, I think, very, very crucial.
Thank you. Our next question. Could the DOE model be integrated with existing knowledge using the Bayesian statistics? And I believe that's for you, Dr. Stosch.
Yes, so Bayesian statistics is, of course, a very interesting concept, because, in fact, we can incorporate knowledge with data, and we incorporate knowledge using a kind of prior that we did use based on the expert knowledge or based on modeling knowledge that we might have. So, yes, this is certainly possible. One can also ask, is it not even better to use Bayesian statistics together with the process knowledge that we have and with the data to not only map out the design space, as was shown, for instance, by Klaus, but also to know what are the limits on our design space? Because, of course, if you're not certain whether you can really work within the area, you're not certain whether you can file this process with the regulators. And so having knowledge boundaries and having boundaries on your design space is particularly useful, and Bayesian statistics can certainly help in this aspect.
Thank you. Our next question is, for hybrid modeling, does the data necessarily be collected from laboratory experiments? Can we use the plant data, and how can we deal with the noise that comes along with the plant data? I believe that'll be for you, Dr. Nargund and Klaus.
Yeah, we can take that one. Thanks for that. Interesting point. So, if you look at plant data today, you know that only a fraction of the measurements are carried out today. So that will change maybe in the future by getting, let's say, non-invasive sensors in place, but still then we will have to deal with a situation where we don't have every measurement that is needed for training and validating twins. So in this case, actually, it will be essential that we develop first soft sensors that are capable of mimicking, let's say, the missing measurements, and this is possible by having, let's say, a digital twin trained, for example, in the upstream development phase. And next, actually, we have to deal with outliers. So, but there are let's say methods around that we can make use of. So this is something we can deal with. I think the most critical point are the soft sensors. So this is a topic that we are also working on, which is essential then also to do the automated control of the processes.
On the back of that, Dr. Stosch, have anything you'd like to add?
Yes, so maybe just adding that we've seen, of course, in the past in literature that people have in fact implied hybrid models using plant data, and for instance, together with Roche, there was a group of Professor Lübbert and Halle-Wittenberg who've shown that using hybrid models with plant data can derive significant benefits for process optimization. So from my perspective, it's all a question of what question and what objective do you want to address when you look at your plant and at your plant data. I think there's certain things that we can directly derive using those data. Also, I agree with Klaus that we have to get much more data to really look into the deep understanding of our process and to derive the deep understanding of our process.
Thank you. Our next question is, other than webinars such as this, can you recommend any specific learning resources to increase the understanding of personnel within pharma companies on the technologies and benefits of hybrid models / digital twins? Klaus and Shilpa, would you like to answer that one?
Shilpa, maybe you can take that one.
Well, fortunately, there are no standard courses on this, or so from my knowledge, I take it as webinars like this or articles that are written on biopharma digitalization or such that can fill in these gaps. We are also trying to generate more information in this field because we do find that the first step for us as well is to in some way educate the personnel in understanding how this is so different from traditional development and what are the advantages, is it advantages or pitfalls and such. And we're generating our own, we're trying to fill this gap, but maybe Klaus or Moritz may have other resources that I may not know, so I would let them answer.
Well, thank you. That's all. Oh, sorry, Jess. Something you want to add?
No, this was already Moritz interrupting in or kicking in. I just wanted to highlight that, in fact, there is a hybrid modeling summer school which is organized every second year, and I think from now on it will be yearly because there's a lot of interest. And this year there's still places available. It's end of the month in Lisbon, so if you're interested, you might want to look out there. We also just published in the article, or I think it's in the process of being published with Biopharma Asia, where we are outlining some of the sources and where we give a better insight into how these models could be designed and addressed and how they can address some of the challenges we face in the biopharmaceutical industry. Also, I've published a book, and this is really on how hybrid models can be developed in the different process industry. I think the book is called Hybrid Modeling in Process Industries, and it was released this or last year. And there's really a lot of chapters, some particular for the biopharmaceutical area, and also on QBD and PUT, where we talk extensively in this book.
Well, thank you for your answers. Thank you for that, Moritz. And thank you, audience members, for your questions. Before I finish the webinar, I would like to ask our presenters, Dr. Stosch, Dr. Mauch, and Dr. Nargund, if you have any closing remarks for our audience. So do you have anything you'd like to add onto that, Moritz?
Well, I maybe just want to add that I think we're living in a very exciting time where, I mean, there's a lot of change happening and there's a lot of opportunity out there, and I feel that many people are scared by mathematics and these kind of things, but I think we should go way beyond this. We should be going way beyond being scared, really take step by step, slowly pace up. And I ultimately think that we can really benefit from the potential that is there and make things happen. And yeah, this is where I also really see myself and where I see my contribution.
Thank you. And Klaus and Shilpa, do you have any closing remarks for our audience?
Yeah, thanks, Stephen. So we are really living in a very exciting time, and 10 years from now, looking back at this time, I'm sure people will be saying that that was the most, let's say, exciting time in the last, I don't know, 20 or 30 years. So actually, we are building up this technology and we are going to apply it, and this digitalization momentum we are only at the starting point, so this will change our world. And we are seeing, actually at Insilico, we're seeing something like a digital bioplant at the end that is a virtual representation not only of let's say unit operations like cultivations, which is then also a representation of the whole plant, including the supply chain, that we can use actually to design new products, new processes right from the beginning, optimize them, controlling them. Having this, let's say, as something like a flight simulator just beside your real plant. So this is actually the vision that we have. That will take some time, but we are going to take a step-by-step approach. And yeah, we'd be happy actually to do this also with our companies that and our partners that bring in such a tremendous let's say momentum into our development.
Well, thank you for that. Thank you for those remarks for our audience. I would like to take this opportunity to thank our presenters. Once again, we had Moritz von Stosch, Senior Manager, Technical R&D at GlaxoSmithKline Vaccines, Klaus Mauch, CEO at Insilico Biotechnology AG, and Shilpa Nargund, Data Scientist and Business Development Manager, also at Insilico Biotechnology AG. Thank you for sharing your knowledge with us. I would also like to remind our audience that you can view this and other webinars on demand by visiting biopharma-asia.com/on-demand-webinars. And if you are watching this on demand, then please feel free to send your questions over to me at stephen.edwards@biopharma-asia.com, and we'll get those answers sent back to you. Thank you, everyone. Have a great day.